Hello and welcome to our discussion on the immune system. We are in Anatomy and Physiology, the second half, and we're now discussing the immune system. In this segment, we're going to talk about some overviews and some functions. We're going to look at some innate defenses, and then later on down the road, we're going to talk about adaptive defenses. That's probably going to be in another segment. This immune system has non-specific and specific defense mechanisms, and it's going to protect us against hostile intruders that enter our body. So perhaps it's worthy to take a second and appreciate that we live in a world full of microbial pathogens. It's noteworthy that we are able to survive with little notice of this potential danger around every corner, so to speak. As we feel comfortable in this perilous environment, notice that we can give a lot of appreciation to the immune system that protects us against infectious agents and harmful substances. These cellular structures we talk about soon, these molecular structures, work together to provide immunity. That is, protection against a specific type of infectious agent as well. That's going to be our adaptive immune system. Here's a nice overview of this immune system, again divided into an innate immunity, which is a wide variety of substances we fight against, and adaptive immunity, delayed response to specific pathogens. It's also noteworthy in this diagram that we have three general lines of defense, each having many important components. Keep in mind that, just generally speaking, these pathogens need to get through two types of barriers in our innate immunity. They have to get through a cutaneous membrane, and they have to get through a mucous membrane. And that is going to do most of the fighting of the immune system. However, if these pathogens can get underneath these membranes, then we have innate defenses that await these microorganisms. And then if these microorganisms are lucky to get past the mucous membranes, the innate defenses, and then that's where the adaptive immune system kicks in. We have cell-mediated immunity, and then we have humoral immunity. So let's take a closer look at this immune system. We'll start by just familiarizing ourselves with some of these things that we're been calling infectious agents. So when we use the term pathogenic, those are agents that can cause harm. Let's look into some of these infectious agents. Our first potential infectious agent are going to be bacteria. Bacteria are going to be microscopic, single-celled organisms that are going to be enclosed by a cell wall. Biologically speaking, we classify them as prokaryotic cells. And most of these bacteria are actually harmless. We go through a daily routine with these bacteria covering our body. However, some of those are virulent, virulent meaning causing a serious illness. Another potential invader to the immune system in the human body are viruses. And it's noteworthy that viruses are not cells. Instead, they're sort of capsules that are composed of DNA or possibly RNA, and then again they're wrapped in a sort of protein capsid. Viruses are obligate, so to speak, intracellular parasites, and that means that they must enter a cell to reproduce. They cannot reproduce on their own. So examples of viruses include the common cold, Ebola, and chickenpox. On looker's right is a histology image showing some of those viruses. And keep in mind these are amazingly smaller than even bacteria. So we need special microscopes to even come close to sensing the general structure of a virus. We don't have a lot of good microscopic images in general of viruses. Another possible foreign invader are going to be fungi. Fungi are eukaryotic cells that have a cell wall. And examples include molds, yeasts, multicellular fungi. These are going to produce spores, and sometimes those spores are irritating. We sometimes get superficial diseases, such as ringworm is a disease on the skin, but we can also get internal infections on the mucosal linings as well. And another possible foreign invader are protozoans. These are going to be eukaryotic cells without a cell wall. And these are a little bit larger than some of the creatures we've been talking about so far. And we also have multicellular parasites. These are visible in a microscope, but not visible necessarily all the time with a naked eye. These multicellular parasites reside within a host from which they take the nourishment from. And another example of a possible infectious host would be prions. These are just going to be fragments of infectious proteins. These are neither cells nor viruses, but they can cause disease in nervous tissue. Take, for example, a quote-unquote mad cow disease. Perhaps one might recall from listening to media that we can spread these fragments of infectious proteins from cows to humans by consuming infected meat. So that was just sort of an overview of some of the items that we are trying to protect ourselves against. So now let's take a look at some cells that are going to help protect us. And one of those cells that protects the human body are leukocytes. We might recall the presence of leukocytes from our discussion in the white blood cell, I should say the blood system. So these leukocytes are formed in the red bone marrow of bones, and these leukocytes include neutrophils, eosinophils, basophils, monocytes. We're talking about T lymphocytes, B lymphocytes, and natural killer cells. We're going to go in depth into each of those a little bit down the road. It's noteworthy that most of these leukocytes are going to be found in body tissue rather in blood, and that's because they can sort of squeeze through some of these larger capillaries sometimes and get to the locations that they need to be. So we have lymphatic tissue. We might recall that from the last chapter we discussed how there's lymphatic tissue strategically placed in regions throughout the body. Last section we talked about things like lymph nodes, the spleen, tonsils, etc. These house the lymphatic tissues containing items again like T and B lymphocytes. And so in addition to finding these cells in lymphatic tissue, we're going to find these cells in certain select organs as well. So that means that these macrophages might be migrating around generally in the body, or they might reside in one particular organ. So let's take, for example, the lungs. We have macrophages that reside inside little pockets inside the lungs. They're going to help kill any any type of foreign invader that might be inhaled into the respiratory system. Speaking of cells, we remember that cells then build up to form tissues. So tissues such as epithelial layers of skin and mucous membranes, those are going to house these dendritic cells. And these dendritic cells are going to be derived from monocytes. They're going to engulf pathogens and then migrate into lymph. And you might recall that once they get into the lymph, the foreign invaders get filtered through lymph nodes. And then connective tissue is throughout the body, and it hosts mast cells. And these mast cells are typically close to the small blood vessels. Here's a couple images just as a review showing the presence of some of that lymphatic tissue we've been discussing. So in summary, this diagram tries to show the location of immune cells. Again, remember that we have a variety of white blood cells, aka leukocytes, that circulate in the bloodstream. We might find many of these in lymphatic tissue, such as spleen, tonsils, malt, etc. Here's an example of a macrophage in the alveoli of the lungs. Here's our example of the dendritic cell that is within the skin and mucosal membranes, our first line of defense, so to speak. And then again, these mast cells that reside within the connective tissue throughout the body. So we're still on this topic of cells and complex molecules that help the body fight foreign invaders, and we get to a topic of cytokines. So these cytokines are going to be small, soluble proteins produced by cells of the immune system, and they're going to regulate and facilitate immune system activity. And we can think of cytokines as a sort of communicator between cells. And that's because these cytokines are chemical messengers. They sort of release a message that sort of binds to the receptors of our target cells. So the effects are signaling the cells, controlling development of immune cells, and regulating an inflammatory response. Also destroying these cells are going to, destroying foreign cells is one of the tasks of the cytokines. These cytokines do have a short half-life. In other words, they break down quickly. That's going to prevent continuous stimulation. We only want these cytokines stimulating when they are needed. So we talk about there being two main branches of the immune system, the innate immune system and then the adaptive immune system. So as an overall guiding factor in this discussion today, we're going to first talk about the innate immune system, and then in another segment we'll talk about the adaptive immune system. So briefly speaking, comparing these two systems within the system, this innate immunity is referring to defense mechanisms of the immune system that we are born with, hence the name innate. The components of the innate immune system respond immediately to potentially harmful agents. And then this adaptive immune system involves specific T lymphocytes and then also B lymphocytes. And these are going to respond to the foreign substances that we're exposed to. This adaptive immunity takes several days to be effective. So even though we have innate and adaptive immune systems that are distinct, they do indeed both work together to provide defense in the human body. And here's a nice summary of that, again, talking about the things we just discussed. So now let's go ahead and discuss innate immunity a little bit more in depth. So this innate immunity includes the body's first and second lines of defense. We sometimes also call this non-specific immunity. So as we said, that's going to prevent the entry of potentially harmful substances. And the first line of defense is going to be the skin and the mucosal membranes. Our second line of defense involves processes such as white blood cells and certain chemicals, and then also the processes of inflammation and fever. And that's where we're going to wrap up today, is around there with inflammation and of fever. So as we said earlier, our skin has epithelial tissue. This epithelial tissue of the epidermis and the connective tissue on that dermis as well is going to provide a physical barrier. And again, that's going to be the main culprit of defense, the main, I should say, soldier thwarting the majority of the pathogens that we encounter on a daily basis. Very few microbes can penetrate this layer. And why is that? It's because the skin releases antimicrobial substances, things such as immunoglobulin A, lysozyme, sebum, and defensins. These are all chemicals that can be released by this tissue. Mucous membranes are going to produce mucus in the body, and that sort of quote-unquote slimy mucus is going to trap microbes. And then just as a general mentioning, we do have other mechanisms of the first line of defense, things like coughing, sneezing, defecation, urinating, vomiting, acidic environments such as the stomach, or secretions of the eyes. These are also other first lines of defense besides the skin that protect us against foreign invaders. So at this point, we're going to go in depth and look at some of those white blood cells a little bit more in depth, these cellular defenses. So coming back a couple slides, here's a histology image showing neutrophils and monocytes. And perhaps you remember these from the unit on blood. Neutrophils are going to be the most prevalent leukocyte in the blood. You might recall that from one of the lab activities we did. And these can morph into macrophages that destroy engulfed particles. And these macrophages are going to reside in tissues throughout the body. These neutrophils and these macrophages are both phagocytic. In other words, they eat sort of invaders, so to speak. Once they ingest or engulf these infectious agents, they subsequently digest these unwanted substances with enzymes from lysosomes. And we might recall that lysosomes are going to be cellular organelles that are in charge of breaking down particles in the human body. Here's a concept called the respiratory burst. That's a mechanism of neutrophils and macrophages, whereas the reactive oxygen molecules are used to destroy the microbes and viruses. Dendritic cells, as we've already mentioned, are going to destroy particles as well. Antigens get presented on the dendritic cell surface to the T lymphocytes. So we'll talk about more of that later on, that quote-unquote presenting. Here's a diagram showing that neutrophil doing its job. Here's the process of a bacteria being engulfed. So here's our infectious agent. Here are those cellular organelles called lysosomes, which break down the substance. And here's the process of exocytosis, the removal of those fragments. Another leukocyte that's going to help out the human body are basophils. Basophils and mast cells promote inflammation. These basophils and mast cells produce chemicals like histamine, heparin, and these are going to promote inflammation. Histamine is going to cause vasodilation and also capillary permeability. Heparin is going to be an anticoagulant, and then, let me pronounce this right, eicosanoids are going to be released from the plasma membrane to increase the process of inflammation. And again, we talk about inflammation towards the end of today's topic. Inflammation is an important part of the innate defense mechanism. Here's some histology showing those mast cells residing within connective tissue. And here's sort of a diagram showing that release of chemicals such as histamines and heparins. And notice how they're having an effect on the capillaries that are near. We can cause vasodilation of those capillaries, thus helping in an inflammatory response. Another important cell in the human body are going to be natural killer cells. These natural killer cells destroy a variety of unwanted cells. Things like virus-infected cells, bacterially infected cells, tumor cells, and cells of transplanted tissue are all generally sort of quote-unquote attacked by these natural killer cells. These natural killer cells are formed in the bone marrow. They circulate in the bloodstream, and then they accumulate in secondary lymphatic structures. It's interesting, their name is sort of unique. These natural killer cells sort of quote-unquote patrol the body, just sort of like a police officer might patrol the streets of Anchorage or wherever. These natural killer cells patrol the body for unhealthy cells. We might also call this immune surveillance, so to speak. And so these natural killer cells release chemicals such as perforin and granzymes that's going to destroy the unhealthy cells. and these granzymes enter the pore and cause apoptosis of the cell. This process called apoptosis is going to be cell death that causes shriveling rather than lysis. And again, here's a natural killer cell attacking a foreign invader, releasing chemicals that sort of quote-unquote punch a hole, so to speak, inside the foreign cell. Here's that process of apoptosis showing the shriveling up, so to speak. Another cell that's going to help the immune system is going to be eosinophil. Here's a histology image showing the cell. And we might recall that eosinophils are going to attack multicellular parasites. These eosinophils target parasites by releasing substances that are lethal to the parasites. Things like enzymes, oxygen-containing compounds, and neurotoxins are going to help fight those foreign cells. So in general, generally speaking, all these cells of the immune system, the innate immune system, are able to recognize foreign microbes because they possess pattern recognition receptors on their cell surface. So these are small, tiny, not even visible with the microscope structures that are biochemical in their nature, and they send a signal to anything that docks or connects with them. So in this diagram, we see a parasitic worm on looker's right. It's larger. We see on the left, here are these eosinophils releasing their cytotoxic chemicals to fight that parasitic worm. In addition to our white blood cells, that is our leukocytes that we just talked about, we have antimicrobial proteins. And these are, such as the name says it, molecules that function against microbes. And these are going to be components of the innate immune system that fight these microbes. Another example is an interferon. And an interferon is a class of cytokines that non-specifically impede viral spread. They complement the other antimicrobial proteins. Here we go. Here we see that these interferons is a class of cytokines, and we can see that they're a sort of non-specific defense mechanism against viruses. So we see here in this diagram that these cells are infected by a virus, so we need to sort of kill that infected cell. We need to interfere with the replication that's going on of the virus inside it. So interferon is going to bind to receptors of neighboring cells, and that's going to prevent them from being infected. And in addition to the prevention, it's also going to trigger the synthesis of enzymes that destroy viral RNA or DNA. And also, this interferon is going to stimulate macrophages and natural killer cells to destroy the virus-infected cell. Other antimicrobial proteins include the complement system. The complement system is composed of plasma proteins, and these are produced by the liver. We're talking about over 30 of these plasma proteins. When we activate this complement system, it involves an enzyme cascade, and we have a classical pathway or we have an alternative pathway. This classical pathway requires activation by the antibody that's going to be bound to the foreign substance, where the alternative pathway is activated without an antibody. The complement system carries out a few processes. We have opsonization, inflammation, cytolysis, and elimination of immune complexes. Let's take a little bit closer look at some of these processes. In the lower part of this diagram, we see the process of opsonization occurring. So this opsonization is a binding of a protein called opsonin to an antigen in order to make it more likely that the antigen will be engulfed by phagocytic cells. Inflammation is going to be enhanced by complement. And so this inflammation is going to activate mast cells and basophils, and it's going to attract neutrophils and macrophages, which we might recall have a phagocytic activity. And other antimicrobial proteins within the complement system include cytolysis. And this is the direct killing of a target cell by forming a transmembrane protein channel. In other words, the attack complex, so to speak. Here we see a special protein placed on the pathogen, and that causes the cell to lyse, or to burst, so to speak. And then also we can eliminate immune complexes. So in Looker's right, we have an immune complex. Notice how there's an antigen attached to an antibody, attached to complement, attached to erythrocyte. So we can eliminate this immune complex, by linking immune complexes to erythrocytes. When we then attach them to the erythrocytes, these become stripped off when the erythrocytes are processed in the liver and the spleen. And now we bring our attention to the process of inflammation. Inflammation, again, is part of the innate immune system. And this is going to be an immediate response to ward off unwanted substances. This is going to be local, non-specific event, and that's going to occur in vascularized tissues in response to some sort of stimulus. Let's go ahead and look at some of the events of inflammation. So keep in mind that this tissue in the human body has received some sort of injury. So let's go ahead and look what happens. Our first step is going to be involving the release of various chemicals. We're talking about releasing chemicals like histamine, prostaglandins, and chemotatic factors from mast cells. And then these chemicals that we just released are going to cause vascular changes. We're talking about things like vasodilation, increase in capillary permeability, and stimulation of the endothelium in the capillaries to produce cell adhesion molecules, CAMs in other words. Another step then is the recruitment of leukocytes. We get a process called margination. Margination is a process by which these CAMs on the leukocytes adhere to CAMs on the endothelial cells of the capillaries. And then in this process of inflammation, we get diapedesis. That's the process by which the cells exit the blood through the vessel cell walls and migrate to the site of the infection. And we're going to see a diagram in a short few showing that activity. Another reaction when you recruit these leukocytes is going to be chemotaxis. That means these leukocytes migrate towards chemicals released from damaged, dead, or pathogenic cells. And they sort of migrate along a sort of chemical gradient, so to speak. That is, chemicals released from damaged or dead cells and pathogens are going to attract these immune cells. Continuing on the events of inflammation, we near the end with delivery of plasma proteins to the site of inflammation. These plasma proteins include immunoglobulins, complement, clotting proteins, and kinins, all get brought to that inflammation site. These clotting proteins sort of wall off the microbes to prevent further spread into blood and other tissues. And these kinins have similar effects to histamine, and they also stimulate pain receptors as well. And in the final process of inflammation, we get fluid called exudate that moves from the blood to the injured or infected area. Exudate is a sort of increased fluid. It's going to also have protein in there and immune cells. And so this exudate is going to sort of flood into the interstitial space. Well, then this exudate gets taken up by the lymphatic system, and that lymphatic system is going to monitor the lymph as it passes through the lymph nodes. And so when we can tell, we can see that inflammation has occurred because we have the redness, the heat, the swelling, the pain, and loss of function of that affected area. And lastly, it's important that we stop the inflammation. This inflammation is going to last about 72 hours, but as we wrap it up, we do not want chronic inflammation. So that means that the monocytes are going to exit the blood. These macrophages eat the bacteria, damaged host cells, and tissue repair begins. The fibroblasts form the new connective tissue. So in summary, we don't want the inflammation process to keep occurring. We want it to end. So here's a diagram showing all those steps that we just talked about. As a quick summary, again, here's our inflammation site, our damage. For example, the child in the upper left has received some sort of cut. Step 1, again, is the release of those chemicals. And then step 2, we get the vascular changes to the capillaries. Step 3, we start recruiting immune cells to the site. And step 4, we have the delivery of the plasma membranes. And then step 5, something not shown, is that we start withdrawing some of these attackers so that the inflammation ends. Let's now turn our attention to the last topic we talked about in the innate immune system, and that is fever. And we can define fever as an abnormal elevation of body temperature, sometimes also called pyrexia. That's going to be an increase of at least 1 degree Celsius from the typically accepted core body temperature of 37 degrees Celsius. In this process of fever, we have pyrogens like interferons and toxins as well from infectious agents are going to produce a sort of fever. And this gets picked up by the hypothalamus in the brain. The hypothalamus is going to raise the temperature, and that's going to lead to a fever. We have stages like onset, stadium, and defervescence. In other words, rising, sort of a plateau, and a decrease. It's noteworthy that fever can result from drug reactions, brain tumors, or sometimes in response to trauma. So during the onset of this fever, we get the hypothalamus is going to stimulate blood vessels in the skin. These blood vessels are going to vasoconstrict, and that's going to retain heat. When we sometimes shiver when we get a fever, the role of that is going to be to increase heat production, and that's going to raise the body temperature even further. During this process called stadium, the elevated temperature gets maintained, and in this process also the liver and the spleen are going to sequester zinc. You might recall zinc as an element on the periodic tables. They're going to sequester zinc during stadium, and that zinc is going to slow microbial reproduction. The process of defervescence is the time when temperature returns to normal. That happens vasodilation in the skin and sweating are going to occur. That vasodilation and that sweating is going to have the result of lowering body temperature. So why might we want the human body to go through this strenuous process called a fever? Well, fever has benefits, and some of those benefits include inhibiting the reproduction of bacteria and viruses. Also, this fever is going to promote interferon activity. It's going to increase adaptive immunity. It's going to increase the presence of those CAMs we talked about. And it's also going to accelerate tissue repair. For that reason, it's generally advised in a medical profession to leave a low fever untreated, because That's a natural process in the human body. However, we do have risks associated with a high fever. And when we say high fever, that's a significant one when it's above 100 degrees Fahrenheit. High fevers are dangerous because of changes in the metabolic pathways. And that's because we can get so hot that we might denature certain proteins in the human body. So we do need to treat a high fever. Seizures may occur at a sustained body temperature above 102 degrees Fahrenheit. And brain damage might occur in body temperatures greater than 106 degrees Fahrenheit. And a death is likely when the human body reaches a temperature of 109 degrees Fahrenheit or greater. So in summary, this fever is a higher than normal elevation of body temperature, and it's part of the innate immune system that's going to help the body protect against foreign invaders. So as a summary of everything we've talked about in this section today, we see a diagram that summarizes innate immunity. This diagram specifically talks about the first line of defense we discussed earlier. So notice in this diagram we see a small child that is sort of mouthing, so to speak, on a toy. Notice how that toy is sort of covered in germs, so to speak. Things like bacteria, viruses are coming into contact with this child's mucosal linings. These bacteria are interacting with the cutaneous membranes within this child. Notice how some of the bacteria have migrated down into the stomach region. This diagram is trying to show that vomiting can help rid the body of some of those microbes. Also, these microbes apparently invaded the large intestines and also the urinary bladder. Defecation and urination can push out these viruses as well. So again, we need to give credit to the skin, mucous membranes as the first line of defense, which thwarts much of our invasion. If they get past that first line of defense, we have a second line of defense in the innate immunity. That's going to be non-specific cellular and molecular defenses. So here we see on a molecular, on a microscopic level, we have things like fever, inflammation, and then other smaller processes. Things like cellular defenses by the leukocytes, and then cellular defenses such as antimicrobial proteins and complement are going to help fight the second line of defense. So in summary, in our innate immune system, we have a first and a second line of defense to help with immunity. Our next section we talk about is going to discuss adaptive immunity, how we have a specific response. At this point, we talk about a homeostatic imbalance. One homeostatic imbalance is an abscess. Abscesses also have this substance called pus in it. So pus is an exudate. That means it has excess fluid, protein, and immune cells. And remember that this stuff is going to leave the capillaries and interstitial fluid. It contains dead neutrophils, and that pus contains damaged tissues, cells, cellular debris, and fluids. So this thing called an abscess is an accumulation of the pus in a sort of walled off area. And because it's walled off, the result is that pus cannot clear. And then we get other homeostatic imbalances, things such as an ulcer. An ulcer is going to be an open sore, and that's typically found often in the gastrointestinal region. People with poor circulation, that is people with diabetes, tend to be at risk for abscesses and ulcers. And sometimes we get stasis ulcers in tissues of legs. That's due to poor oxygen and nutrient supply to tissues. So again, quite often, if we cannot beat the condition with the genetic conditions given, if we improve our diet or possibly improve our lifestyle through exercise, we might thwart this homeostatic imbalance. So that does it for the innate immune system. We're going to take a break, and we're going to start a new section here in a few minutes, and that's going to be called our adaptive immune system. Talk to you then.